Chapter 6 will detail information regarding specific nutrient requirements as well as the growth requirements of organisms. There are different types of nutrients that a microorganism requires. All organisms require some form of a nutrient source. Those that are absolutely required by an organism can be classified as essential nutrients and can be further broken down into macronutrients and micronutrients. The macronutrients are ones that the organism requires in large amounts and are necessary for both structure and metabolism. Carbon, hydrogen, and oxygen fall under this category. Micronutrients are also required, but in smaller amounts, and are useful in enzyme function and maintaining proteins. We can also classify nutrients based on their carbon content. Inorganic substances are those that do not contain carbon or hydrogen, although an exception being carbon dioxide. These include metals, salts, gases, and water. Organic substances are those that contain carbon and hydrogen and can be as simple as methane or something as complex as a protein. The majority of the cytoplasm consists of water. In addition to water, we find an abundance of proteins. A significant amount of the dry weight of the cell comes from organic substances and carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. E. coli contains roughly 5,000 different compounds, but it only needs to absorb a small amount of nutrients to synthesize them. In these tables, we can see the major organic and inorganic compounds found in E. coli. We can also see carbon, hydrogen, oxygen, and nitrogen making up the majority of the dry weight. When it comes to microorganisms, the world is a buffet for them. Let's first look at the carbon source for an organism. Those that are heterotrophs get its carbon in an organic form. An autotroph uses carbon dioxide as its inorganic carbon source. These types of organisms do not depend on others for nutrients. We can then classify organisms based on their energy sources. Those that are photosynthetic are termed phototrophs, while those that obtain nutrients from a chemical source are chemotrophs. This particular table outlines the categories of organisms based on their energy and carbon sources. This is a great study tool to help you prepare for your exam. Let's look at the different autotrophs. Photoautotrophs are organisms that can perform photosynthesis and use inorganic substances like carbon dioxide to produce their own organic substances. Chemoautotrophs can be separated into two groups. Chemoorganic autotrophs use organic substances for their energy source and an inorganic substance as their carbon source. The lithoautotrophs do not utilize sunlight or organic substances. Instead, they rely on inorganic substances like hydrogen sulfide. Let's now discuss the heterotrophs. The chemoheterotrophs rely on organic substances for both their energy and carbon source. They use these substances in cellular respiration or fermentation, depending on the organism. Saprobes are the decomposers who feed on dead organic material. Parasites are those organisms that feed off of a living host. Pathogens, those that are capable of causing disease, are considered parasites. They can live in a variety of conditions, whether it be on the body, in organs, or inside of cells. There are some parasites that require a host cell for survival, such as a virus and the Bacillus bacterium associated with leprosy. This table, along with the next ones on the next couple of slides, outline where the essential nutrients can be obtained. In addition to the essential nutrients like carbon and hydrogen, microorganisms also depend on other nutrients. Potassium is vital in protein synthesis, sodium is important for transport, and calcium helps to stabilize both the cell wall and endospores of a bacterium. So we know that they must eat like you and I, but how do they do it? They transport the substances directly across the cell membrane. The tendency of atoms and molecules being in constant motion is what drives the movement of the substances. Diffusion is the movement of a substance from where there is more of it to where there is less of it. Osmosis is the diffusion of water across a semi-permeable membrane. By being semi-permeable, there are openings for water, but other substances cannot pass. Water will continue to move across the membrane until equalization has occurred. Here we can see the red dots representing a solute and the blue ones representing water. We can see the semi-permeable membrane, which has pores that are small enough to allow water through, but prevent the passing of the solutes. There is a large concentration of water outside of the sac, therefore the water will diffuse into the sac. In an isotonic solution, there is an equal concentration of solutes inside and outside of the cell. In this scenario, there is an equal flow of water into and out of the cell, so nothing happens to the cell. In a hypotonic environment, there is a greater concentration of solutes inside the cell, which means there is more water outside of the cell. The water rushes into the cell and causes the cell to swell and potentially burst. In a hypertonic environment, there is a larger amount of solutes outside of the cell and more water inside. So, water moves out of the cell via diffusion. This causes the cell membrane to separate from the cell wall in a process called plasmolysis. Passive transport, like diffusion and facilitated diffusion, do not require energy. Active transport utilizes some energy source to move substances. Again, active transport requires energy and transports substances against the concentration gradient. Amino acids, phosphates, and metal ions are some examples of substances transported via this mechanism. Endocytosis is the process of bringing a substance into a cell with the help of a vacuole. Phagocytosis is a process seen in immune system cells and amoebas, and is the process of bringing a solid particle in. Pinocytosis is cell drinking, and is the process of bringing liquids into a cell. Every microorganism has preferred temperatures at which they grow. The minimum growth temperature is the lowest temperature at which an organism will grow. The maximum growth temperature is the highest temperature in which growth will occur. Above this, metabolism slows and proteins become denatured. The optimum temperature is the temperature that is just right. It's the temperature that will give the fastest rate of both growth and metabolism. Let's look at the classification of organisms based on their temperature requirements. Psychrophiles are those that prefer cooler temperatures. The best temperature for them is below 15 degrees Celsius, but they can still grow at 0 degrees Celsius. Most of the time, we put our food in the refrigerator, but for these organisms, that only enhances their growth. We could find these guys in lakes, rivers, polar ice, and even in the deep ocean. The psychrophiles are rarely pathogenic because our body temperature is too hot for their survival. Psychrotrophs can grow in the cold, but their optimum temperature is between 15 degrees Celsius and 30 degrees Celsius. These guys are classified as our spoilage bacteria. Organisms like Staph aureus and Listeria can grow in the refrigerator and cause foodborne illnesses. The mesophiles are our pathogens. They grow best between 20 degrees Celsius and 40 degrees Celsius. They can be found in plants and animals, and in soil and water. Their optimum temperature just so happens to be our body temperature. Thermoduric organisms can survive in high temperatures for a short period of time. We encounter them in contaminated heated or pasteurized products. Your endospore formers are classified as thermoduric. Thermophiles grow above 45 degrees Celsius and can be found in areas associated with volcanoes and compost piles. The majority of eukaryotes can't survive above 60 degrees Celsius. You also have extreme thermophiles that can grow in excessively hot environments. This figure does a great job at allowing you to visualize the differences in the various organisms based on temperature requirements. Your blue dots represent the minimum growth temperatures, while the red represent the maximum growth temperatures. All microorganisms have gas requirements. Oxygen is the most important when it comes to microbial growth. It's great for respiration, but is also a strong oxidizer. As far as organisms go, there are some that can both use oxygen and break it down. There are those that can't use it, nor can they break it down. And last but not least, those that don't use it, but they can detoxify it. Oxygen exists in various forms. A singlet oxygen is highly reactive and can oxidize the lipids and completely damage the cell. Superoxide ions are also reactive, along with hydroxyl radicals. Hydrogen peroxide is also toxic to cells. The majority of organisms have developed mechanisms to neutralize these toxic molecules. The first reaction is to break down the superoxide ion into hydrogen peroxide and water. And get an oxygen. But it doesn't stop there, because hydrogen peroxide is also toxic and must be broken down. That's where the catalase enzyme comes into play. It breaks down the hydrogen peroxide into water and oxygen gas. Let's look at the classification of organisms based on their oxygen requirements. Aerobes use oxygen for metabolism, and they have the enzymes necessary to break down the toxic molecules. Those that cannot grow without it are considered obligate aerobes. There is special media which has oxygen at the top and an anaerobic environment at the bottom of the two. This allows us to classify organisms based on their growth patterns. Aerobes, like Mycobacterium tuberculosis, would grow at the top. Microaerophiles are those that can't survive in high levels of oxygen, but do require a small amount. H. pylori which causes stomach ulcers, which show growth between the top and the bottom. Facultative anaerobes don't need oxygen, but if you make it available to them, they will use it. Staphylococci species will grow throughout the entire tube, with a heavier presence in the aerobic environment. Anaerobic organisms do not possess the enzymes necessary to break down the free radicals of oxygen. Bacteria that we find in our mouths fall under this group. Obligate anaerobes will only grow at the bottom. Aerotolerant anaerobes, like Streptococci species, don't necessarily use oxygen for metabolism, but they do have enzymes that allow for their survival in the presence of oxygen. They will grow throughout the tube, but typically more towards the bottom. Capnophiles are those organisms that thrive in high levels of carbon dioxide. Organisms like Neisseria gonorrhea must be isolated and immediately incubated in a carbon dioxide-rich environment, or they will not grow. The pH scale runs from 0 to 14, with 7 being neutral, below 7 being acidic, and above 7 being alkaline. Most microbes prefer to grow in a pH between 6 and 8. Now there are those that love the acidic habitats, and those are your acidophiles. Euglena, which is a protist, can grow in a pH of 0 and 1, which is even more acidic than a car battery's acid. Your molds and yeast typically love acid habitats. On the other hand are the alkalinophiles, with archaeal species growing at a pH of 12. Proteus, a bacteria that causes urinary tract infections, does so by neutralizing urine, which has antimicrobial properties. Your osmophiles love habitats with high solutes, while your halophiles love salts. Facultative halophiles like Staph aureus can grow in moderate salt conditions of 0.1 to 20%. There are special media used in the laboratory to grow these species. If an organism is not a phototroph or photosynthetic, light tends to damage them unless they have specific pigments. UV radiation is used to decrease the amount of microbes on surfaces. Barophiles survive in a significant amount of pressure. Sometimes we can find microbes in shared habitats. We may find them living with the same microbes or with plants or animals. Sometimes the microbe does nothing to their roommates, although they can be harmful. Most of the time, they cohabitate for nutrients. A symbiotic relationship is one in which two organisms live in a close relationship. In a mutualistic relationship, two organisms live together and both benefit. An example of this would be the organisms that live in and on us. They get the nutrients that they need, and in turn, we get development of our immune system. Commensalism is a relationship where one benefits and the other is unharmed. Parasitism is a relationship in which the parasite, like a virus, gets nutrients and a habitat, and it harms the host in the process. Antagonism is when we see competition between organisms. They're competing for space as well as nutrients. Antibiosis is a situation where a microbe produces substances like antibiotics to kill its competition. Of course, we capitalize on this. Synergism occurs when two organisms who work together come to a common goal and both benefit. bacteria working together in our mouths may lead to gum disease and cavities. You've seen the term biofilm before. This is a community of bacteria that attach to a particular structure. One guy comes in and attaches, calls all his friends in, and then further creates this dome, if you will, that protects the community from our immune system and antibiotics. Here you can see, once they attach, they secrete this extracellular matrix that also further allows for attachment. Once established, the bacteria can break free and eventually form even more biofilms. This image shows the bacteria in blue attached to gauze fiber in green. The orange represents the extracellular matrix. The bacteria that we find forming biofilms have different genes than what we see in other microbes. Let's discuss how bacteria replicate. They do so through a process called binary fission. In the process, one cell becomes two. Let's look at this process. We start off with a young cell. We can see the cell wall, chromosomes, and ribosomes. The first step is the enlargement of the original cell. We can see the duplication of both the DNA and the ribosomes. This is to ensure both cells have an equal amount. A protein band then develops in the center of the newly enlarged cell. Eventually, a septum forms, and we see the separation of two cells. Each cell is identical to one another. All bacteria are different. Some may undergo this process in 15 minutes, while some may take hours. The generation time is how long it takes for a bacteria to go from one cell to two. A doubling effect occurs constantly, provided that the conditions are favorable. Again, this generation time is different for all bacteria. The majority of pathogenic bacteria and those we study in the lab have pretty short generation times. This image shows the doubling of cells with each generation, and we can see the dramatic increase in the number of colony-forming units in just a matter of 20 minutes. A growth curve shows us the pattern of growth for a particular bacterium. The bacterium is initially inoculated in a sterile broth, incubated, and sampled at specific intervals. The samples are then plated, and the number of colonies are counted. If done correctly, we should see a dramatic increase within a matter of several hours. Let's discuss this growth curve and its stages. The lag phase is a period of adjustment for the bacteria. This is where they kind of figure out what nutrients are available and how to utilize them. There's not a period of substantial growth here. In the log phase, this is where we see rapid or exponential growth of the bacteria. This will continue so long as nutrients are available. In the stationary phase, there's an equal amount of birth and death rates for the bacteria. Cell division starts to plateau as with the growing number of bacteria, nutrients and oxygen are being depleted. The death phase is where we see death of the bacteria at an exponential rate. More cells are dying than births. A viable non-culturable state describes how there are cells that are alive in the death phase, but kind of hide out so they don't grow on media and are not counted. Here we can see our curve. Lag phase being that adjustment stage, so no rapid multiplication. Exponential or log phase is where we see rapid growth. The stationary phase is where we hit a plateau as far as growth is concerned. And finally, our death phase, where more cells start to die, but we do also have live ones in there as well. Turbidity, as you know from your lab, can also be used to assess the general concentration of bacteria. The more cloudy or turbid the broth, the more bacteria. A spectrophotometer can be used to determine turbidity based on the absorbance of light. We can also do direct cell counts to determine the population size. A Coulter counter is great, but it does not differentiate between live and dead cells, so it counts both. A flow cytometer can detect differences in live and dead cells. Genetic testing can also be done. Here we see a direct count in which bacteria are placed on a slide and microscopically examined. Problem with this is human error, and our eyes can't tell the difference between live and dead cells. Here we can see the Coulter counter, which electronically counts, but again, can't differentiate between living and dead cells.